we do have some new information with the announcements, so I'm gonna wait until 8 o'clock before we do that, and we'll make sure everybody's here for the new information. But won't hurt to remind you guys the same stuff I say almost every day. So again, you can retake exam one as many times as you want for the practice of it. It'll help you prepare for the final exam. Yeah, but it's too late to change your grade for exam one. Now, you still can meet me online, get answers for exam one. Again, you'll need that to help study for your final exam. So in addition to getting the the words, brief. In addition to getting the correct answers for exam one, just like any other assignment that you need before, you'll get a 5% boost to your grade. So that's exam one. Any questions about exam one? All right. Exam two. Still, you can retake it, even if you've already I cannot speak today. Even if you've already retaken it and I've adjusted your grade, you can still retake exam two. Just like usual, you'll email me when you're done, and I'll go in there and adjust your grade. And to remind you, with these retakes, there's nothing to lose. If for some reason you somehow get a lower grade, despite the fact that it's overbooked and not timed, and you can retake it as many times as you want, we'll just keep your original higher grade. So, nothing to lose, retake these exams. So that'll close when exam three closes, which we'll talk about here in a couple minutes. Yeah, so I will say this, I'm probably preaching in the choir, because you guys who are here don't generally have issues, I don't think. But every semester, at the end of the semester, somebody's going to be concerned with a grade, and they're going to ask me, is there anything I can do about my grade? I can't get whatever grade that they're probably about to get. And I should have been saying this throughout the semester, but I'll say it now. This is what you can do about your grade. Things like this. So please don't come to me at the end of the semester. And again, I'm preaching in the choir, but I hope somebody will watch the video later. Please don't come to me at the end of the semester asking if there's anything you can do about your grade. If you didn't do things about your grade throughout the semester, did you retake the exams? Did you correct your labs? Did you meet me online to get feedback and a 5% boost? These are the things you need to do to prepare for the exam. Or excuse me, to get a decent grade. Now's the time to do it. And know that I'm here for you, right? I'm here to help you. So that's it for the reminders about exam one and exam two. It is 8 o'clock, so I will go ahead and discuss exam three. So we are almost done with chapter 9, which means we're almost done with the material for for exam 3. I think we should be able to get it knocked out at least by Friday, meaning we can take the exam next Monday or next Friday. So your first word for attendance is either Monday or Friday. You choose. So that's your vote. The first word for attendance is a vote, whether or when you would like to take the next exam, next Monday or next Friday. There's advantages and disadvantages, right? If we take it on Monday, the material will have been fresh in your head and you'll have all weekend to study for it. But then again, if we wait until next Friday, then you'll have more time to study for it. But the disadvantage is by then we'll have started the material for the final exam, so it could get a little bit confusing. But anyway, yeah, that's what exam 3 is. Monday or Friday, you vote. That's your first word for attendance. It's going to cover chapter 6, 7, 8, 9. And we'll have to meet here in person just like we did for exam 2. So be prepared to meet here in person. Now, if you don't want to meet here in person or you know you can't, contact me between now and then. I'm more than happy to meet you online and proctor your exams so you don't have to come in. And so we can extend the time limit, right? You have 50 minutes to take the exam when you come in here on that day. But if you meet me online, we can essentially eliminate the time limit. Plus, you can ask me any questions that you might have about the questions. So, any questions about exam 3? All right, nice and easy. So remember, again, your first attendance word is going to be either the word Monday or Friday. Underline those for people who are watching the video and need to take a screenshot, or anybody who's watching the live stream. So that's it for the exams. Any questions before I take attendance and then continue on with the announcements and report? All right. Oh yeah, one more thing really quickly. Unfortunately, we don't have time to do like reviews, official reviews, where we take a lecture, an entire lecture and review for the exam. So another thing you can do in your email, and this isn't for attendance worth it, somewhere in your email when you send for attendance, if there's a day and time that you would be willing to meet online to do a review where I'm looking at the exam and I give you the answer, you won't see it, but I'll say some version of the question out loud so you can write it down and have some version of the question and we can talk about the correct answer, all that. I would like to do a review for the people who want to do it. So if you have a date and time when you think that would work, let me know and I'll see if I can make that happen for you. Anyway, so that's it for exams. Let's do attendance really quickly. Ashley is here. Barrett is here. Wooten is not here. Trahan is not here. Dickens is not here. Clark is not here. Ika is online. Isaiah is not here. Smith is not here. Watson, not here. Kaylee is here. Lucas is not here. Marley is here. Noah is here. Savannah is here. Mia is here. Edwards is here. Wilkes is not here. Did I miss anybody? Nice and easy. Okay. Speaking of being here, and going back to the announcements and reminders, today's lab, even though it is virtual, is going to be a team effort. So unless I've left feedback, and some of you, at least one of you who are in here, unless I've left feedback that says you don't have to be here for lab today, then you'll need to be here for lab today. Make sure you have your computers. For anybody watching this who's not going to be in lab today, you're going to have to jump. You'll either only get half credit for missing the lab, or I'm going to have you jump through some extra hoops. Like some of the extra credit will not be extra credit for you; it'll be required. But anyway, any questions about lab today? All right, attendance. I'm not going to repeat that. It's the same stuff I always say, right? I'm going to do the attendance words. You're going to write them down if you're watching the video, or if you're watching the live stream, you're going to take a screenshot. You're going to email all that to me, along with one question about the material, right? One question about the material. And then, of course, if you're watching the video, you're going to also like or comment on the video and include that screenshot. Anyway, let's jump into it, back into it. So we're almost done with the second main bullet point, which will put us about 2/3 way done with this chapter. So again, like I said, this is the last chapter that's going to be on exam 3. So once we're done with this and we start talking about viruses and things like that, then we'll be done and we'll be ready for exam 3. So let's talk about mutations. The mutations are important. We're going to talk about mutations now, and also we're going to talk about them in the next chapter. So mutations will be on this exam because we're talking about them now, and they'll be on the next exam because we'll be talking about it, for the next chapter, and because the next exam, the next exam after exam 3 is the final exam. It's got about 100 questions. About 50 of them are from the first of the exams, and about 50 of them are from the last chapters. So it's a double reason why it's going to be on the next exam, right? Not only will it be on the new material, but it will cover the first 3 exams, and this will also be on the first 3 exams. Anyway, a mutation is any change in the nucleotide sequence of DNA. So this is different than what we talked about with genetics, right? This is not epigenetics. Epigenetics was like, well, the code is the same. Again, using the same analogy that I've been using for a while. You've got these books of instructions, right? That's your chromosomes. Books of instructions about how to build proteins. Epigenetics would be more like something happened to the book where it's harder to open it up to that page. Or maybe the pages are stuck together. Or kind of the opposite. Maybe the book just flops open constantly to a certain page, right? So something happens to the book where it's harder or easier to express these genes, to read the chapter. Mutations are different. Mutations is like going in there and editing the book, right? That is what I mean by any change in the nucleotide sequence of DNA. To remind you of stuff that we've talked about previously from this chapter that you're going to need to know for the exam, this language, this nucleotide language, consists of 4 letters, right? A, C, T, or G if it's DNA, A, C, U, or G if it's RNA. And every word in the language is 3 letters long, right? So just remind you of what we're talking about. So again, a change in the nucleotide sequence means we're changing the letters in those words. Now, there can be big ones or small ones, right? You can involve large regions of chromosomes, single nucleotide pairs that occur in sickle cell. We'll talk about that. And again, it's just an example of the sickle cell, but that is a great example. And anything in between, right? So big, small, anywhere in between. Does anybody have any questions about what a mutation is before we start talking about the different types of mutation? All right, so this picture is not from your textbook, I don't think. But you notice here it says, here's the original sequence. It says point mutation. We haven't talked about what a point mutation is yet, but we will. But really quickly, just to show you where this looks, talk about this image. Here we have a T and a T as it should be, an A and an A, an A and an A, a C and a C. Here, in the original sequence, we're supposed to have a T, but it's been changed to a C. That's a mutation, right? Well, one type of mutation. It's a very small mutation, but it is a mutation. Here's another example. This is from the old textbook. Again, not from your textbook. But this is not some hypothetical example like the last one. This is a real example. Not that you need to memorize anything, again, because this is just an example. But this is where sickle cell disease comes from. You have this whole gene, and when one letter is wrong, in this case, it causes sickle cell. So instead of having this a codon that says CTT, instead, there's a codon that says CAT, which results in, instead of having this GLU amino acid, you have this VAL amino acid. And so that one small change is enough to completely deform the protein, which is then enough to deform the entire cell, which then is enough to cause this entire disease of sickle cell. So anyway, any questions about that example? It's nothing to memorize. Again, it's just an idea of what a mutation can be, and how even a very small mutation can be catastrophic. All right, so we have different ways of categorizing these mutations, and you do need to know this for the exam. The first way we're going to categorize them is there's either going to be nucleotide substitutions, which is like the one we just saw, right? Instead of having an A, I think it was a T, whichever. But again, just an example, so that exact letter doesn't matter. The point is, that last example I showed you for sickle cell was a substitution, right? Instead of having this letter, we have that letter. That's one category, and we're going to talk about substitutions separately. And then we have nucleotide deletions or insertions, which again, we're going to talk about separately. So deletions or insertions is one, or you could do deletions / insertions. That's one category, and the other category is substitutions. In other words, either we change a letter, or more than one letter. Either we're changing letters, or we're changing the number of letters, right? So we're either adding or taking away, or we're editing, changing, replacing, however you want to put it. The next slide is another example of a substitution mutation. Just like the last one I showed you about sickle cell, this is another example. Here, instead of having a GGC, we now have AGC. So instead of getting the GLI amino acid, we get the SER amino acid. Again, just an example. You don't need to memorize any of those codons. The point is, we've changed the letter, and therefore we've changed the amino acid. Now, we're going to talk a little bit later, just so you know, sometimes you can change a letter and not change the amino acid. Because if you remember that big chart that I showed you, that big circle chart, sometimes there was more than one codon that coded for that particular amino acid. So sometimes, again, you can change the code, you can substitute something, but still end up with the same amino acid. And again, we'll cross that bridge when we get there. It would still technically be considered a substitution, but there's also another category that we're going to put that into. And we'll talk about it soon. So any questions about substitutions or this slide in general? All right, here's an example of a deletion. This is pretty simple. We've simply, you can see, instead of having a U, we pulled out that last U. And this is the bigger deal. And you can't really see it much on this screen because we're only seeing a very short portion of the gene. But think about this. If this was written like to scale and the whole gene was written out, I bet it would go all the way around the room, right? If we could do that. So what's happened here is once we've done that, everything this way shifts, right? And again, it looks short because, I mean, we have a little bit of information, a little bit of space, but imagine if that was going all the way around the room, all those letters, all of those will have shifted, meaning you didn't just change one amino acid now, you changed all the amino acids, right? Everything downstream from there, you've changed the amino acid. So obviously, deletions are a much bigger deal than substitutions because you're changing a lot. Unless, of course, somehow you had the deletion was at the very last codon of the protein. It'd be less of a big deal then. It would still be a big deal though, because you would be changing at least one amino acid. And as you know from sickle cell, changing one amino acid can be a big deal. So that's it for deletions. Any questions about deletions before we talk about insertions? All right, insertions are just as catastrophic. Again, instead of having a U, we've now inserted a G in between those last two U's. And again, just like in the last example, you can't see it, but imagine what you've done now since you've inserted something, you've shifted everything that way. So now everything downstream has been changed. So this is a huge deal, right? And just so you know, and I'm going to talk about it more officially later, but I just want you to start thinking about these things now. When we start talking about evolution and natural selection, it's all because of stuff like this. Without mutations, we wouldn't have natural selection. There would be no diversity within organisms, right? There'd be no variety. And natural selection works on variety because this variety works better than that variety. And nature chooses one of those. Anyway, that's the insertions. Any questions about insertions? All right. Now, this part, hopefully this is pretty simple to follow. But unfortunately, when we talk about mutations, we have all these different ways of categorizing them. So here's another way of categorizing mutations. We have missense versus nonsense, and you will need to know the difference between those. The missense mutation changes the amino acid coding. So everything we just talked about so far, those have been missense mutations, right? We have changed the amino acid sequence either by substituting one for another or by inserting one and then also shifting everything downstream, screwing it up, or taking one out and again screwing up everything downstream. Those are called missense mutations. Then we have nonsense mutations. They're simple concepts, but I can imagine trying to memorize these for the exam could get a little bit confusing because they sound similar. But a nonsense mutation is basically it's instead of coding a codon coding for a particular amino acid, you change it to a stop codon. One of the test questions about that is going to be something like, what would happen if you had a missense mutation? That might be all it says, I don't remember. And the answer is basically you would end up with a shorter polypeptide, right? If a missense mutation changes a regular codon to a stop codon, right? So even if everything downstream from it is still fine, it doesn't matter because you have that stop codon. So to remind you how translation works, once the cellular machinery gets there, when it's in that ribosome, once the ribosome recognizes that stop codon, it says, oh, our job is done. And it just releases everything. The polypeptide goes one way, the mRNA goes one way, the two subunits to the ribosome go their own way. Right? So when you've stopped, you would have a smaller polypeptide. All right, so those are two things. This should almost be on different slides, right? So these two bullet points to go together. The next ones are a little bit different. You also need to know what a frame shift mutation is. And again, we've already talked about it, but now I'm just giving you the word for it. So when we have an insertion or a deletion, that is called a frame shift mutation. Hopefully you can see why, because like I showed you, the entire thing shifted. Right? When we took one nucleotide out, everything downstream shifted. When we inserted one nucleotide, everything downstream shifted. And that's why they call it a frame shift mutation. I've already mentioned that they can have disastrous effects, right? Because again, it's not just one amino acid you're changing, it's everything downstream. And usually it does produce a non-functioning polypeptide. I'm probably not going to ask you anything about that, but it's just worth noting that that's usually what happens. Yeah. But again, sometimes it's good. Sometimes, this is how we have new proteins, because we have big mistakes like this, right? Big mutations will cause new proteins. And sometimes those new proteins, instead of being non-functioning, which they usually are, sometimes you get lucky and it's a very beneficial protein. Like let's look at the moth, for example, since you guys have already done that lab. I don't know if it was just one mutation. Well, we know it was a mutation, right? I don't remember if it was one mutation. If it was just one protein, is what I'm saying. But let's just assume it was for this discussion. So they had this huge change, right? So now instead of having a light-colored moth, now there's a completely different phenotype, the dark-colored moth. And that was advantageous, right? So this is the kind of things that are important to keep in mind as we move forward past exam 3 and start talking about natural selection. Anyway, the next word for attendance is down there at the bottom. You can just abbreviate it HC. It's Harvey Cushing's birthday. For extra credit on a different email, you can look him up. Tell me why how he could be relevant to biology. Don't forget to include your source if you look anything up and send me an email for extra credit. Also, for those of you who have done it, you already know this, but to remind everybody else, since it's extra credit, it's not like top of my priority list. Right? I have a lot of things I need to get things graded on time. So extra credit stuff, it'll get graded eventually, I promise, but it might take me a while to get to. Anyway, any questions about what's on this slide before I show you another example of a frame shift mutation? Okay, then. Here's another example of a frame shift mutation. It's the same thing I showed you better, but in a way, this is kind of, it's the same thing I showed you earlier, but I think it's better because at least it's longer, so you get more of a sense of what happens when again, it's kind of hard to see, but you can see we have all these codons. Each codon is blocked off. And what we've done here is we've shifted it, right? Each one has been shifted by one. So now again, everything downstream is a new amino acid, which is the big deal. We're not changing one amino acid, we're changing all the amino acids. That is a frame shift mutation. This is also not from your textbook. It's a great studying tool, this little chart. It's got everything in one, everything we just talked about in one table. So if you don't, if you want to study this, you need to download the PowerPoint. So again, we have the two different types of mutations. You have a substitution versus an insertion or a deletion, and then we can look at their effects. Oh, one thing I didn't mention officially, so here it is in writing now. I mentioned it, but here it is in writing. A silent mutation results in no changes to the amino acids. So like I was getting at earlier, sometimes you can change a letter, but the codon is still the same in a sense. Right? The codon, just remember, some amino acids have more than one codon for that amino acid. So sometimes you can substitute a letter, right? You can substitute a nucleotide and get a different codon. Since that codon codes for the same amino acid, you still get the same exact polypeptide, the same exact protein. Therefore, that is called a silent mutation. So there, that is in writing. You need to know that. But we've already talked about this one. We have missense and nonsense. And of course, when we're talking about insertions and deletions, they all result in a frame shift mutation. Any questions about this? I'm hoping that the content is simple and easy to understand, and hopefully it's just the nomenclature that you have, maybe that's all the challenge is memorizing that it's this word for that thing and this word for the other thing, but hopefully the concepts are easy to understand. All right, let's talk about what causes mutations. These are mutagens. Mutagens, again, these are things that cause mutations. They do this in a number of ways. Sometimes it's stuff we've already talked about. So you learned about how DNA duplication works, right? You know that DNA polymerase attaches to the double helix and opens it up so it can read the code, and then it brings in the complementary nucleotide and adds it one at a time. So you have to imagine sometimes mistakes happen. As a matter of fact, one of you asked about that on one of your questions for the attendance. And generally speaking, it does not happen, right? So generally speaking, if you have the wrong amino acid, the wrong nucleotide, it's either going to be too big, right? Because remember, double, some of them are double, some of them are single. It's either going to be too big or too small. And another thing, like I mentioned, that you don't need to know for the exam, but just to remind you, some of them have 3 hydrogen bonds, some of them have 2 hydrogen bonds. So there's fail safes to keep it from happening, but it does still happen. So again, you could have an error in that situation. Or of course, recombination. So we talked about sometimes when a chunk of a chromosome will come off and be inverted and come back onto the chromosome, or sometimes a chunk of a chromosome will come off and go to a different chromosome. Of course, then there's also sometimes crossing over. And we know that happens a lot. But think about this. If you had a crossing over event in the middle of a gene, you messed up that gene, right? So this is, again, how things can happen like that. Not much we can do about that. More common for us, though, are physical and chemical agents. So the most common physical mutagen is high energy radiation. And most of that comes from X-rays or UV light. And this is why they say, protect your skin. This is another great example, by the way. A lot of people don't seem to understand this concept. There is such a thing as too much of a good thing, right? We need light. Having sunlight is healthy for us. We need it for vitamin D. Obviously, the world needs sunlight because that's where we ultimately get all of our energy, with a few exceptions. But again, too much of it, too much of specifically the UV lights, not necessarily the visible spectrum, but that ultraviolet light is high energy, and they can go in there and mutate our DNA. So I just want to point that out because there's a lot of fear mongering from people who don't understand science and biology. So someone might say, you know, I've seen it too. Someone suggests, hey, protect your skin, whether that means wearing a big hat or, you know, clothes that cover your skin or sunblock. And people are like, you need sun. Like, yeah, you do. But we're getting plenty of, most of us are probably getting plenty of sun. So, you know, when you find yourself outside on a bright sunny day, it's good to protect your skin. Anyway, those are physical mutagens. Chemical mutagens, you can look those up for extra credit if you want. Again, don't forget your sources. There's all kinds of chemical mutagens. Usually when someone says things like, oh, that increases your risk of cancer, that's usually what they're talking about is a chemical mutagen, right? Smoking. There's some chemicals in that cigarette that, you know, can mutate your DNA. We have a lot of stuff in this building locked away safely that we use in some experiments. Just be very careful with your gloves because they are mutagens. They could mutate your DNA. And as you know, sometimes mutations can mess up those proteins that are involved in your cell cycle. And we know that when that happens, that could lead to cancer. So it's a good time, too, to remind you of all this. I should have said that at the beginning, but here we are talking about mutations. And remember, it's basically, I'm oversimplifying, but basically it's mutations that cause cancer, right? We mutate those genes, therefore we don't have the proper proteins that are in control of the cell cycle, and then that's what will get cancer. Anyway, any questions about this slide? Okay, actually, another thing I'll point out that you don't need to know for the exam, but it is worth mentioning. I remember, speaking of chemical mutagens, I don't remember what it was that they were talking about, but I saw something that said, you know, if you eat, I think it was either like sausages, or maybe it was the grilled stuff, like the burnt stuff on grilled meat. One of those two. I just remember it because I like both of those things. Anyway, they said something like, if you eat this, it doubles your chance of getting cancer. And that sounds scary, but then I looked into it, and whatever it was, let's just say it was sausages. And if you eat sausages, like however much a year or a week, whatever, then it increases your risk of cancer by 1%. No, what was it? Maybe it was grilling the sausage. Anyway, let's say eating sausage, and there was something like a 1% chance of getting cancer because of you eating the sausage. So then when they say you grilled it, it doubled that. That sounds scary, but all that means is you went from 1% to 2%, right? If you double 1%, it's 2%. So nothing to worry about for the exam, but again, some common sense that people seem to lack because they don't understand basic science. There we go. Still talking about mutagens here. Again, very often mutations can be harmful, but sometimes they're very useful in nature. So we've already talked about natural selection, right? Sometimes these mutations cause new proteins that are actually working proteins and helpful proteins. And sometimes we even take advantage of this fact. Like, for example, for my thesis work, we were trying to figure out which genes are responsible for trichomes on plants. Trichomes, little hairy things on them. So there's a lot of ways of doing that. Once we've narrowed down the ones that we thought were the ones that were in charge of trichome growth, some of the things we could do with them is mutate them. All right, if these genes truly are responsible for protein or trichome growth, then if we mutate them, then something should be different about their offspring, right? When we look at the new plants that come from those mutations, they should either have a lot more trichomes or fewer trichomes or fatter trichomes. Like, something should change about the trichomes if those genes are responsible. So that's how we can use mutations in the library. Not that I'm in the laboratory. Not that I'm going to ask you that on the exam. So again, mutations are the source of diversity. That will also be a test question for the next exam. Not on this one. But, again, once we start talking about evolution and natural selection, it's all about that variety. Variety is the nature of choosing one variation over the other. And without mutations, we don't have variations. So anyway, Any questions about that slide or about mutations in general? Because we are now done talking about mutations. Okay. So that means we're done, 2/3 done with the chapter. Finally, we can get into this last portion where we talk about viruses and other non-cellular infectious agents. The next word for attendance will just be IS instead of the full Izzy Stradlin. This person has nothing to do with biology, so I'm not going to give you extra credit for looking them up. It's just a musician from Guns N' Roses. Anyway, let's talk about viruses. Again, one of you asked a question, a good question recently on the attendance emails. And I mentioned something about viruses and how technically they're not alive, right? They do share characteristics of living organisms. For example, they have genetic material in the form of nucleic acids. So do we, so do living things. That genetic material is packaged in highly organized structures, just like ours. But they're generally not considered alive because, first of all, and this goes way back to chapter 1 when we talked about the properties of life. They're not cellular, and that was a property of life, right? If it's not cellular, it's not life. And it can't reproduce on its own. It basically hijacks your cells, and your cells do the reproduction for it. And that's what we'll be mostly talking about when we talk about viruses. We'll be talking about how they do that. So yeah, for the exam, make sure you know that viruses are similar to living things in that they do have genetic material that's highly organized, but they are not alive because they don't reproduce and they're not cellular. At least that's what the old textbook says. I don't remember what your textbook says. When you think about it, there's other things too. Like, for example, since they're not cellular, the viruses, they can't even, there's no homeostasis, right? They cannot control their environment. You know, we had a whole discussion about how cells can control what comes in and out of the cellular membrane, and they wouldn't be alive without that. Viruses can't, right? They don't have a cell membrane. They do have a membrane sometimes, at least have a protein coat. We'll talk about that in a second. But that's it, right? They're not maintaining any pH. They're not maintaining any, concentration of hydrogen ions like we do in photosynthesis and respiration. They don't do any of that. So all that, again, just falls under the whole idea of not being cellular. They don't have cells, so they can't do all that. Any questions about this slide? All right, a virus is an infectious particle consisting of genes in a box, basically. What I mean by genes in a box, I mean nucleic acid wrapped in a protein coat. As you're going to learn here in a few minutes, it can either be DNA or RNA. But either way, it's some sort of nucleic acid. It is wrapped in a protein. And in some cases, you'll see there'll even be a membrane. Like our cells have a cell membrane. Sometimes they have a cell membrane. Of course, even in that case, even the ones that have a cell membrane, within that cell membrane, there's still it's still wrapped in a protein coat. So there'll always be a nucleic acid wrapped in a protein coat. Sometimes it'll also have it, basically, I hate to call it a cell membrane. By definition, it's not because it's not a cell, but it is a phospholipid bilayer. Again, a virus cannot reproduce on its own. This is how we get infections, right? Because they hijack your cells. And now that you've learned about how transcription and translation work, hopefully this should make sense to you, right? So, for example, again, the virus could be DNA or an RNA, depending on the type. If it was a DNA virus, if it could get its DNA into your cells and trick your cells are just going to read that gene, right? Oh, there's DNA. RNA polymerase is going to go up to it. It's going to read it. It's going to make an mRNA, right? That mRNA is going to be eventually translated into the proteins. And of course, the virus could be an RNA virus where it does give you the mRNA. So it's like, all right, here's some mRNA. And if it can trick your cells into doing it, your cells will be like, oh, there's mRNA. I better translate that into a protein. So far, I've been pretty mostly repeating myself. But anyway, again, viruses aren't alive. They can't reproduce on their own. But they do consist of nucleic acids. Any questions about this slide before I show you a generic picture of some protein or some virus? I think this is from the old textbook. All right, any questions? So here you can see there's the protein coat and there's the nucleic acid on the inside. In this case, the nucleic acid happened to be DNA. Also, there's these little spikes that hasn't even been labeled. But we've talked about the spikes before too. Not all of them have spikes. Some of them do have spikes. They all have different purposes. And you can look it up for extra credit. But I was mentioning when we talked about the mRNA vaccine for COVID-19, that's how it works. It doesn't give you the dangerous stuff, right? There's nothing about that. It's not like, all right, here's the genetic information for this virus. It's not. It's just like, here's the code so they can build this protein spike. Because the protein spike by itself is harmless, right? So then your body builds it, then your immune system recognizes it and learns how to fight it. That way, when you do get exposed to the virus, your immune system has already learned how to fight it. Sometimes that means you won't get it at all. Sometimes that means it'll reduce how bad you get it. And sometimes it's just not that effective. A lot of things to consider there that we don't even talk about in this class. Anyway, that's just a picture. Let's move on. Now, there's different types of viruses. I'm going to go through this pretty quickly because for the most part, that's in the name. I bet if I were to test you right now, you could probably guess which is which. So if I said, which of these viruses do you think infects plants? Oh, maybe plant virus. Which one do you think infects animals? Oh, an animal virus. Which one of these do you think affects specifically only humans and not other animals? HIV, right? You could probably guess on this. But anyway, for each one of them, there will be some new concepts. So it's a little bit less important to realize which ones attack which. But in the process of talking about how they do it, you can learn how viruses work at the molecular level. So again, the first one we're talking about are these things called bacteriophage or bacteriophage. It's in the name, right? They eat bacteria. These are things that infect bacteria. They're often called phages for short. They consist of DNA enclosed in a structured protein. This is a good picture of your typical phage. It almost looks like a space capsule. It looks pretty futuristic to me, which is kind of crazy when you think about it, because viruses have been around for a long time, longer than humans. But anyway, yeah, that's what one looks like. It's a good time to remind you, nowhere in this exam, I don't have any plans of showing you a virus and asking you what kind of virus it is. So don't worry about that. Will be good extra credit question, though. And ironically, the most complicated looking virus, anyway, this one, is the one that attacks the most basic life forms, right? So this attacks bacteria. Then later, I want to show you what an animal virus looks like and a plant virus. And it does not look as complicated. Of course, sometimes those can be deceiving. But anyway, any questions about this side before I show you another picture of some phages attacking the bacteria cell? All right. This is an actual scanning microscope picture. It's zoomed in. What we have here is the bilayer, the phospholipid bilayer, the membrane of the bacteria cell. Here we have 3 different viruses that have attached. You can see this one's squirted and starting to squirt in its genetic information, its DNA. So is this one. This one's coming in by far. Looks like that one just started. Anyway, also, again, I'm not going to ask you their size, but yeah, that one's 200 nanometers tall, just so you know. But yeah, there'll be no questions about the images. But yeah, that's how it works. And of course, as always, for extra credit, anything I say, I invite you to look it up to see if it's true. You know, hey, you said this, and according to this source, it agrees with what you said. Or, as always, you can dig deeper. Like I said, that the DNA is leaving the virus and entering the cell. But I didn't talk about the mechanism of how. How's that working? Does it get sucked in from the cell? Does it get pushed out from the virus? There's some sort of chemical reaction. Is it a diffusion thing? So you can look it up for extra credit. Anyway, here's some more pictures. This is a better picture, I think. I mean, it's a drawing, so it's not as cool, but it's better at visualizing so you can see what's what. See, this was called phage lambda. Not that you need to know that. This one is attacking this bacteria. This shouldn't make much sense to you yet, but we'll talk about how it works in the next few slides. And it's basically like a little hypodermic needle, right? It gets in there, squirts in the DNA, which is what this red stuff is. And you can see here, this green stuff, as you're going to learn later, that represents the genome of the bacteria. So that's the DNA that it already had. And what this is doing is it's inserting itself into that genome. right? So you can see this little bit of red inserted into that circular chromosome. Bacteria have circular chromosomes. And now, every time that bacteria were to reproduce, of course, this particular one's not because it's dead. We'll talk about that in a second. But if it were to just stay in that way of being, then it wouldn't die right away, right? But every time it reproduced, when the DNA polymerase goes in there and reads the code and does a perfect copy of it, it's also going to copy that viral DNA as well. So it's in there forever. But anyway, eventually, sometimes, as you're going to learn in the next slides, sometimes because that is incorporated into the DNA, once that DNA is transcribed, then we start having the mRNA that gives the instructions on how to build the virus, the new virus, right? So now this cell is building a bunch of new viruses, so many, in fact, that eventually the cell explodes, basically, and it releases all those new viruses that now go out and infect another cell or other cells, plural, and the process continues. So anyway, everything I just described here doesn't write it. You're going to need to know the difference between the lytic cycle and the lysogenic cycle. And I've just described the lytic cycle and the lysogenic cycle. I just didn't use those words. So I'm going to use those words now, and then a couple of slides, I'll show you another visualization that'll make a lot more sense because it'll be labeled and it'll really help you. But yeah, lytic cycle and lysogenic cycle. Before I even talk about it, I'll give you a little hint. This isn't good for necessarily learning the concepts, but a hint for the exam. So if I was a tutor, this is the kind of information I would give you. When I describe them, you're going to learn one of them is a really short cycle. Like the lytic cycle is very short. It's not much to describe. It's kind of bam, bam, be done. The lysogenic cycle is longer and more complicated. So that's how I remembered it as a student, was the one with the longer, more complicated name just also happened to be the more longer, complicated process. The one with the shorter, easier name is the one that has the shorter, easier to understand process. So lytic versus lysogenic. Yeah, so here it is in writing, but again, I'm going to say this all again when I show you the image. It'll make a lot more sense then. So the lysogenic cycle involves, this is exactly how I showed you on the previous slide, that viral DNA incorporates itself into the bacterial DNA, right? But it doesn't produce new viruses yet. Therefore, it doesn't kill the cell yet. The cell is allowed to reproduce, right? And that's part of the lysogenic cycle. That's part of why it makes it longer. Lytic cycle, it just kills the cell right away. Bam, you're done. Here, again, it gets reproduced. The bacteria can reproduce over and over. So now you're going to have all these new bacteria. But then when something triggers it, then we had translation, and that's, or excuse me, transcription. And that's when we read that bacterial DNA, that viral DNA that's incorporated in the bacterial DNA, and we create that the virus. Just so you know, the word for that, and I haven't used this word yet, so here it is. I've described it, but I haven't used the word for it. The name is prophage. So when I talked about that red bacterial DNA, that other picture, and I showed you how it had incorporated itself into that green circle of DNA from the bacteria, that was what's called prophage. It's when the viral DNA incorporates itself into that chromosome, that one chromosome that the bacteria has. Right? The bacteria only have that one circular chromosome. Anyway, any questions about this slide before I show you a picture of what we're talking about? Another picture. All right. Also, if you download the PowerPoint, you can watch an animation that shows it, the lytic cycle. You can watch this animation that shows you both the lytic cycle and the lysogenic cycle. But now here's the pictures. Lytic cycle, like I said, short and sweet. It's very simple. The virus, you can see here, there's the bacterial DNA. That big circle is the one chromosome. The virus inserts its DNA, but they stay separate, right? So we have that viral DNA and the bacterial DNA. They haven't incorporated themselves, they haven't merged. So right away, that viral DNA is transcribed and translated, and it builds new viruses. And eventually it builds so many new viruses that the cell explodes, and the word we call that is lyse, but you know that from the previous labs. So in lysis, it explodes, and now we have all these new viruses out there ready to infect more bacteria. So then this one goes on and infects a different bacteria cell, and the cycle continues. That's the lytic cycle. Any questions about the lytic cycle? Okay. Like I said, lysogenic, longer and more complicated, right? So still, we're starting off the same. We still have a virus attacking a bacteria cell. It's still inserting its DNA into the bacterial cell. But you can see here, eventually, that viral bacteria incorporates itself, that viral DNA incorporates itself into the bacterial DNA. And now we have what's called a prophage. So now every time this thing goes through mitosis, right, every time it reproduces, it's making a perfect copy during S phase of interphase. So just to remind you of that, right? So we keep making copies and copies and copies, all these new bacteria that have that viral DNA, right? So we have a ton of them. And eventually something triggers it. We haven't got there yet. So yeah, that's how it works, right? That's how the cycle continues. That's the lysogenic cycle. And then we bring them all together. Eventually what happens with the lysogenic cycle is something triggers it. And then these cells, these bacterial cells that are in the lysogenic cycle that have that prophage, eventually they do go into the lytic cycle where that DNA is transcribed and then translated. And then we build more viruses. And then again, the thing explodes and the cycle continues. So yes, lytic cycle is different than lysogenic cycle. Lysogenic cycle is longer because by definition, it's just reproducing, right? It's surviving and reproducing. It's not until something triggers it where those viruses that were produced via the lysogenic cycle can then enter the lytic cycle. That's where they die. So for extra credit, another thing you can look up look into is how, like what is it that triggers the bacteria to go from the lysogenic cycle to the lytic cycle? What causes it? Anyway, any questions about bacteriophages? That's the last we're going to talk about that. Next thing we're talking about is plant viruses. I feel like we can knock this one out in 5 minutes and be done until 9 o'clock for lab, for those of you who need to be here. Plant viruses, pretty simple. They attack plants. Not that I'm probably not going to get this deep on the exam, but just so you know, they usually don't kill plants. They usually just stunt plant growth, which is useful or important for us because a lot of times we rely on plants for food. So if it stunts the plant's growth, that means it could also diminish crop yield. So this is why it's relevant to humans. Most plant viruses are on RNA. I don't know, if I were to ask you anything on the exam that's on the slide, it would be that bullet point. Most plant viruses are RNA. I'm not going to ask you anything about TMV, but we'll talk about it anyway. A lot of plant viruses, including TMV, have that rod shape. You can see up there at the top right. It has a spiral arrangement of proteins, which you can see on that picture, surrounding nucleic acids. And you can barely see that, but inside here is a little bit of red, right? So inside that spiral, inside that cylinder, inside that rod is the nucleic acid, usually, again, RNA. So TMV specifically, not that I'm going to ask you, but this is the example that your old textbook uses, so I kept it in there. TMV infects tobacco plants, obviously hence the name, but also related plants, like tomatoes are actually related. And it causes discolored spots on the leaves. And I think the reason why it's often used as an example is because it is the first virus that was discovered. And once again, for extra credit, you can look into that too. Who discovered it? When did they discover it? How did they discover it? How else is TMV bad? Like, is it that bad to have discolored spots on the leaves, or does it do other things that are worth mentioning? Things you can look up for extra credit. All right, here's a picture. Again, this is from the old textbook. That's a tobacco leaf with miscolored tobacco leaf. Unless you're familiar with what a regular tobacco leaf looks like, and that probably doesn't mean much to you. This is the TMV virus, and then your little square, because we're zooming into that, and this is what it looks like. You can see the spiral protein encapsulating that spiraled up RNA. So exactly like I explained it. So your old textbook also included this, but this is not an agriculture class, so I'm not going to ask you any of this. But I'm just including it just to give you the information. A plant virus has to get past the plant epidermis, right? That's the issue. Just think about a plant doesn't have a respiratory system. Like that's a lot of times how humans will get a virus, right? Because we breathe it in. And the plant doesn't necessarily eat right? It creates its own organic material, so it can't get a virus that way. So yeah, it has to get through the epidermis. That means the damaged plant is more likely to be infected. That's how it's going to get in. Also, sometimes insects will carry a virus because a lot of times insects, that's how they get their food from a plant. They're getting through the epidermis. And of course, farming tools, right? So if you're out there cutting leaves or trimming things, if you hit one plant that has a virus, and then you whack another plant and break the epidermis, then you're going to spread the virus. There's also no cure for most viral plant diseases. So for that reason, agriculturists basically look for ways to prevent it, and also to prevent, breed varieties of plants that are basically immune to it. The next word for attention will be focus. Focus is the next word for attention. I mean, man, I cannot think today. Focus is the next word for attendance. Hopefully I can snap out of it before 9 to do this loud. Anyway, that's it for plant viruses. Any questions about that? We had a little bit more to say about animal viruses, so instead I'll just really quickly, since we only have like less than a minute, let's talk about, I'll give you the introduction, and then when we come back on Friday, we'll finish the discussion. Obviously, animal viruses are common causes of disease for animals. There's obviously other causes too. The old textbook points out that so far, even with COVID, in the grand scheme of things, in the big history of humans, flu has been the greatest health threat than any other virus. And like many other animal viruses, it has an outer envelope of phospholipid membrane. It has spike proteins that I've already talked about. And just so you know, the membrane, the fact that it has the membrane isn't trivial. The fact that it has a phospholipid bilayer, which is basically like a cell membrane, allows it to enter and leave the host cell. Notice the plant viruses didn't have that, which makes sense. Because remember, and I didn't mention this when we were talking about that, but plants have cell walls, right? Animal cells don't have cell walls. So you just merge right in with the cell membrane. And that's it. When we come back on Friday, we'll talk about that. And if you need to be in lab, we'll start at 9 o'clock. And if you don't need to be in lab, I will see you on Friday. And don't forget, when you send the emails for the participation points, don't forget to ask one question about the material. Watching the video, don't forget to like or comment on the video and take a picture of that. Provide even more evidence that you actually did watch the video.